I welcome the students, scholars, researchers, and scientists all over the world. This is Advanced Multiple YouTube channel. Advanced Multiple Incorporation actually a knowledge processing and information management organization that is located in Mississauga, Ontario, Canada. My name is Dr. Taha Nazir. I'm working as scientific executive with Intellectual Consortium of Drug Discovery and Technology Development Incorporation. Okay, well, I'm going to share my information, my knowledge, and my research under the title of Synthesis and Clinical Use of Monoclonal Antibodies. For this purpose, I collected the scientific information and research data from different sources like PubMed, Medline, Scoopers, Google Scholar, National Library of Medicine, MBAS, DOAJ, Directory of Open Access Journal, Boroquist, CNKI, that is China-based database company, stand for China National Knowledge Infrastructure. Some information are collected from ResearchGate and CrossWeb. Okay, well, before moving forward, I would like to talk a little bit about antibodies. An antibody or immunoglobulin produced by a single clone cell called as monoclonal antibodies. It is Y-shaped protein produced by the beta cells, and the immune system identify and neutralize the foreign object. Foreign object means that maybe the virus, maybe the bacteria, or maybe any toxic material or any protein within the human body. Antibody are secreted by a type of white blood cells called as plasma cells. Monoclonal antibodies are antibody identical because they produce from single parent cell. Polyclonal antibodies are derived from different cell line and differ in amino acid sequence. Okay, well, there are the three main type of the monoclonal antibodies on behalf of their structure or the function. I mean, like monoclonal antibodies are divided into three main categories because of their chemical structure, their molecular feature, or their function. Okay, well, the first one is murine source monoclonal antibodies. Second one is the chimeric monoclonal antibody, and third one is the humanized monoclonal antibodies. So the murine source monoclonal antibodies, these are rodent monoclonal antibodies with excellent affinity and specificity generated using conventional hybridoma technology. Clinical efficacy comprise of the HAMA. HAMA means human anti-murine antibody response, which lead to allergic or immune complex hypersensitivity. Chimeric monoclonal antibody, the chimers combine the human constant region with the intact rodent variable region, affinity and specificity unchanged. It also causes human anti-chimeric antibody response. Humanized monoclonal antibodies continue only the CDR of the rodent variable region grafted onto human variable region framework. Okay, well, this is very simple, regular overview of the cell and antibody mediated immunity. We know that God granted us a defensive system that comprised of the cell and antibody. We can divide it into the two parts. First one is the cell mediated immunity, and second one is the antibody mediated immunity. We are not talking very much about cell mediated immunity, but we are going into the depth of the antibody mediated immunity. And we know that whenever antigen means it may be the virus, maybe the proteins, maybe the bacteria, or maybe the any foreign entity, when it enter into the human body capture it. Which part capture it? APC. What is the APC? APC means antigen presenting cell. Antigen presenting cell actually comprise of the two main cell. The first one is the macrophage, and second one is the dendrite cell. These APC actually captured the foreign entity, virus, bacteria, antigen, and process it, and then produce the epitope. This antigen that is converted into the epitope. The epitope is then given to the T helper cell. T helper cell receive it through the TCR. TCR means T cell receptor, and at the APC and attach like major histocompatibility complex. Major histocompatibility complex is one of the protein that is located at the surface of APC. This HMC attached with the TCR of T helper cell to exchange the epitope. When epitope is identified and recognized by the T helper cell through the TCR, T cell receptor, then it is activated. When it is activated, it starts producing the interleukin 4, interleukin 5, and interleukin 2. So interleukin 4 and 5, they actually activate the beta cells or the B cells. B cell then converted into the two part or two type of the cell. The first one is the plasma cell and then memory cell. Memory cell actually store the information and the data, and the plasma cell they activate it. They start producing and synthesizing the antibody. On other side, interleukin 2, they actually activate the cytotoxic cell. The cytotoxic cells start producing the cytotoxin. Cytotoxin means the chemicals or the different kind of the mediators, modulator. They are able to destroy the cell and damage the cell. So this interleukin 2 cytotoxic cell most of the time used for the cancer cells. So this is the major mechanism by which antibody produced in the human body. Now we are going to talk a little bit about the structure of the antibody. Structure of antibody comprised of the two main chain. The first one is the light chain and second one is the heavy chain. They attach each other with the disulfide bond. And there are the two region of antibody. The first one is the variable, second one is the constant. Constant region actually provide the integrity, provide the shape, and it keep it in its actual form. And variable form, that is variable all the time, that is changed all the time. Why it is changed? Because it have to identify and attach with the different all different kind of the toxins, all different kind of the antigens. So the variable part of the antibody that is changed all the time with accordance of the situation to capture and identify, attach and kill the all different kind of the antigens. And the constant region that will remain constant all the time. Okay, how monoclonal antibody are synthesized? This is very simple diagram highlighting and illustrating the synthesis of monoclonal antibodies. We get the antigen. What is antigen? Antigen maybe the bacteria, maybe the viruses, maybe the any different kind of the protein. It is injected to the mouse. Mouse become immunized. It's beta cell activated. They start producing the antibody. These activated beta cells are then fused with the tumor cells. We know tumor cells having the competency to produce all different kind of the proteins, and they are hyperactivated. They actually enhance the unnatural growth. So the speed of synthesis of enzyme and uh, selection of the particular protein mean antibody, they merge together two different features, two different characteristics. They fuse together to produce the hybridoma. The hybridoma then start producing this uh, proteins. Then we need some particular type of the antibody. How can we identify it? How can we sort it? There are different technology that are used to identify the exact type of the hybridoma that we need and desire in clinical or scientific purpose. They are then further cloned, expanded to produce the monoclonal antibodies on commercial scale. Okay, well, this is another diagram that actually highlight the synthesis of monoclonal antibodies. Mouse is activated or like stimulated or immunized by the antigen. Its spleen cell isolated. They are fused together with the myeloma cells, the cancerous cells. These hybridoma cells are then cultured in the hat medium, and then desired particular hybridoma cells are isolated and sorted. They are then eventually harvested for the particular monoclonal antibodies. Okay, well, this is another diagram that highlights the synthesis of monoclonal antibodies. Very simple antigen isolated, identified, and injected to the mouse. Mouse is become immunized. Its beta cells are activated. These beta cells are then fused with the myeloma cell to produce the hybridoma. So these fused cells then cultured in the hat medium. Hybridoma are then screened by the ELISA. ELISA mean enzyme linked immunosorbent assay, and we then have to identify the hybridoma capable of producing the antibody of interest. So these antibody of interest then selected hybridoma producing antibody of interest, and antibodies producing hybridoma clone further for producing in large scale. These hybridoma are then expanded for the production of the antibody in a commercial large scale to pack for the commercial use in a clinical setting or in hospitals. Okay, well, this is the very simple diagram highlighting the 7 usage of the monoclonal antibodies in different industries. The first one is the first type of the monoclonal antibodies is the glycomodified monoclonal antibodies. Second one is the alter amino acid in constant region. Third type of the monoclonal antibody is use different human monoclonal antibodies isotope. The fourth one is link isotope to monoclonal antibody with stable linker. The fifth one is link drug to monoclonal antibody with cleavable linker. The sixth one is insert DNA from monoclonal antibody variable region fused to signaling peptide into the T cell to the to reduce expression of the CAR. The seventh one is cross-link region from the two monoclonal antibodies. Okay, well, this is very simple diagram that actually highlight how the cancer cell can be killed by using the monoclonal antibodies. There are the 7 steps involved in killing the cancer cell. The first one is the release of the cancer cell antigen, then cancer cell antigen presentation to the dendrite cell to produce epitope present it to the T helper cell, then priming and activation, trafficking the T cell to the tumor, infiltration of the T cell into the tumor, and the sixth step is the recognition of the cancer cell by the T cell. At the final and last step, the killing of the cancer cell. It is very simple and regular mechanism by which cancer cell can be used by using the monoclonal antibodies. In conclusion, the future development of the prospective research opportunity may help to design effective immunotherapy drug. The current health policy may support and formulate specific technical guideline in view of the characteristic of monoclonal antibody drug pharmacology, clinical research, and quality management. Additionally, it is recommended to explore the patent term compensation system and award incentive for researchers for development or the developing the innovative system. So the monoclonal antibody drug innovation policy analysis and investment management should be specifically studied in future research. Before moving forward, I would like to salute and greet to my colleague and my coworker like Sherry Bajkar, Wolfgang Dance, Daniel, Dustin Hawker, Marwa Al Maghribi, then Robert Heyman, Kunza, Nudin, Faisal, Terry, Istiaq, Arslan, and Amin. They are my people, my community. They helped me a lot to make this video today. I also acknowledge the kind contribution of Advanced Multiple Incorporation and the Intellectual Consortium of Drug Discovery Technology Development Incorporation. Thank you very much for watching the video and staying with me for so long time. Take care.